Thank you, Petronella, for this amazing introduction. It's really a pleasure to be here. I'd like to thank you and Natalie for the invitation to present today. So this is a nice title, but that's not exactly what I'm going to talk about, even though it's related. So as Petronella just mentioned, I'm working on the HIV reservoir and on the memory subset in which the virus is able to persist during antiretroviral therapy. So a lot of the assays that I'm going to use and present today have already been presented actually by Petronella and Uwe in the two previous talks. We are measuring integrated HIV DNA. We are measuring viral production from sorted cell subset. So I'm not really going to detail the experimental design of the experiments, but more the big idea. So this is the clinical definition of the viral reservoir during antiretroviral therapy. And what happens is that if you start, if you start antiretroviral therapy, your viral load will go down quite rapidly and will reach undetectable levels by weeks and will remain undetectable as long as you will take your medication. But if for any reason you stop therapy, then the virus comes back. And so that's the evidence that the virus is still persisting somewhere in some cells and probably in some tissues when viral load is suppressed. So during these past few years, and I should say starting in 1997 by the group of Bob Silicano and Doug Richman and also Tenue, we know that HIV is able to persist during antiretroviral therapy, and there are several mechanisms that contribute to HIV persistence. The first one is the possibility that the virus still replicates at low levels, and this is most probably true in tissues. If you look at lymph nodes or gut, you might be able to find some new events of viral replication because antiretroviral drugs might not optimally penetrate in those sanctuaries for the virus. But more importantly, or I should not say more importantly, I should say additionally, the virus is able to establish a latent reservoir in some specific cells. The most studied and obvious subset in which HIV will establish latency is CD4. That doesn't mean that HIV is not also able to persist in other cell types. So in the blood, it's pretty clear that HIV latency exists, and you can evidence it quite easily by just getting CD4 T cells from virally suppressed donors. You put them in culture, they won't produce virus, but if you activate them in some ways, then you will see replication in the supernatant. And those cells are able to persist through two main mechanisms. The first one is T cell survival. Those cells are long-lived, so once they're generated and once the virus is integrated within the chromatin, they can persist for decades. But also homeostatic proliferation, which is a natural mechanism that contributes to the persistence of memory CD4 T cells. So that's the way we would like the reservoir to be, which is like a lot of CD4 T cells with a few integrated genomes in some of these CD4 T cells. And we have a tendency to consider that all the reservoir cells are the same, but in fact, they are definitely not the same. And this is coming from the diversity of the memory CD4 compartment. We know for many years now that CD4 T cells come in very different flavors. And here is a cartoon summarizing what we know based on the classification of the memory CD4 T cell subset from the naive cells that have not seen their cognate antigen to those very terminally differentiated CD4 T cells that are relatively short-lived. We have this variety of memory CD4 T cells, those stem cell memory that have been recently discovered, the central memory that are long-lived, transitional memory that proliferate at low levels, and effector memory that have more effector functions. And these are the different features of these cells. And as you can see, there is kind of a gradient in their capacity to produce cytokines, to survive, and to self-renew. So the experiment we did with Rafik in Montreal back in 2009 was to sort the different memory CD4 T cell subset and measure integrated HIV DNA to try to identify the long-term reservoir for HIV. And these experiments were done in individuals who have been on suppressive therapy for at least 3 years. And what we found, and here I'm showing the contribution of each subset, is that the central memory cells were the major contributor to integrated HIV DNA, which is the assay we used in this particular study. But you could also find integrated events in transitional memory and in effector memory, and also in some cases in the naive and terminally differentiated cells, but their contribution was much more limited. More importantly, it was not only integrated HIV DNA, but we were actually able to grow virus of these cells by putting them in culture and activating them. We were able to show that the virus can actually replicate in these different subsets. So the first question I'd like to, or the first data I'd like to show you are the one we've generated in collaboration with the military HIV research program in the US, with Jean Tenat and Anne Varonich. And basically, what the hypothesis we were testing is that by treating or by giving antiretroviral therapy very early on, we could prevent the establishment of the reservoir in those different memory CD4 T cell subsets. So very early means that you have to catch those patients who have been very recently infected. And to determine for how long they've been infected, you can use this classification that has been developed by Mike Bush and one of his postdocs named Fibig back in 2003, which is based on the positivity of different clinical tests from viral load, antigenemia for P24, then the first antibodies, etc. And then you can say that the Fibig-1 subjects have been infected for approximately less than 17 days, Fibig-2, 22 days, etc., etc. So based on this, again, clinical data, you can try to get an estimate of the date of infection. So that was very convenient, and that's the thing we used in this study, which is called RV254 or Search10, which is conducted in Bangkok in Thailand. And basically, these are young MSM who are highly exposed to HIV, and every 3 days they go to the clinic, and we are taking, or they are taking a drop of blood. They are pulling all the blood together, run an HIV RNA test, and if it's positive, then they call back the patient, they convolute, identify him, and what is remarkable is that they're able to start antiretroviral therapy within 2 to 3 days after diagnosis. So that's really the best way, and as Petronella said earlier, maybe an utopia, because we don't use those tests in the clinic every day, but that gives us the advantage of testing this hypothesis that very early ART would prevent the establishment of the reservoir. So these are the data in the blood, and I think the data speak for itself. So these are people who have started antiretroviral therapy during chronic infection. So at day 0, everybody starts ART. Those people in black are already chronically infected, and you can see that antiretroviral therapy has a very minimal impact on the frequency of cells harboring integrated HIV DNA. And in contrast, in people who are starting out at the Fibig-1 stage or the Fibig-3 stage, so very early on, less than a month, less than 2 weeks approximately, you see that the reservoir decreases very rapidly to reach undetectable levels in most of the Fibig-1 that we've looked at. So going back to the CD4 T cell subset, we sorted them because we had leukapheresis from these patients. And here I'm showing you the data of the reservoir after 2 years of therapy. And what you can see is that in the chronic subjects, obviously the reservoir was detected pretty much everywhere. In the Fibig-2 to 5, I would say it's half and half. But in the Fibig-1 subjects that we sampled, we were unable to detect a single trace of integrated HIV DNA, suggesting that initiating ART within the first few weeks of HIV infection is able to dramatically reduce the size of the reservoir. That doesn't mean that it's not there, it means that it's greatly reduced. Okay, so I just showed you the study we did in Montreal, which is basically people who started ART during chronic infection, were followed for 2 to 7 years. And I told you that the reservoir is mainly in CM, TM, and EM, central memory, transitional effector memory. I also showed you this data from Thailand, from the RV254 study, in which patients started out very early on and were treated for also 2 to 7 years, and we are still getting samples from this. But another category of patients you would like to look at, and because now we're starting to have access to these people, are those who have started ART more than 15 years ago. So antiretroviral therapy, the triple therapy, was implemented in 1996-1997. So now we are able to get samples from these people who have been on ART for a very, very long time. So in addition to the side effects, of course, we were interested to the reservoir and the localization of the reservoir. So that's a study we conducted in collaboration with, that is funded by AMFAR. This is conducted in collaboration with UCSF, Steve Diggs, Sarah Palmer in Australia. And we recruited 6 patients who have been on ART for more than 15 years. And again, we sorted the different subsets, and this time we also sorted the stem cell memory cells that have been described quite recently. And what we found is that still, after 15 years of ART, integrated HIV DNA was still detectable in central memory, transitional memory, and effector memory cells with a lower infection frequency in the naive and stem cell memory subset. So integrated DNA is great, but not enough. Again, a lot of these genomes are actually defective, so you want to show that they are or not inducible. And for that, we developed an assay that we called TILDA that measures the frequency of cells with inducible HIV. So the readout we're using here is our multiply spliced HIV RNA, which are usually not produced in cells that have defective genomes, and we're using a 12-virus PMA Enomycin to generate this. And this assay has been developed by Francesco Procopio at VGTI. So we used TILDA in this cohort of 6 subjects to have been treated with ART, and again we were able to find that, yes, those subsets still have the ability to produce virus, or at least multiply spliced HIV RNA, with the effector memory cells showing the greatest ability to produce those transcripts. So here I'm showing you the same data in a different way, which is the relative contribution of each subset to integrated HIV DNA, and pretty much as we saw in Montreal, the central memory cells seem to be, I mean, there are some exceptions, obviously, but seem to be the major contributor to the HIV reservoir. But if you look at TILDA, the picture is quite different. And we found that actually the contribution of effector memory cells seems to be larger for the inducible virus, suggesting that those effector memory cells actually harbor a larger fraction of the real virus after very prolonged therapy. Actually, if you do the ratio between integrated DNA and TILDA to get an idea of the inducibility of these genomes, you would see that effector memory cells harbor genomes that can be induced between 10 and 20% of the time, whereas for the central memory cells, we are between 2 and 3%. Okay, so obviously, after describing those reservoirs, the most important question is, can we interfere with HIV persistence in this memory CD40 cell subset? So a very popular way to interfere with HIV persistence is what we call the shock and kill strategy. So basically, you would take a molecule that would force viral gene expression in those latently infected cells, which will lead to the production of viral proteins and probably viral particles as well. But because this cell is not going to be visible to the immune system, if you have a good immune response, this cell will be eliminated. And of course, the idea is to keep antiretroviral therapy during the shock and kill strategy, so the virus that is produced cannot reinfect new cells. And actually, this strategy has been tested in vivo in at least 3 published studies, one by Dave Margolis 2 or 3 years ago now, more recently by Sharon with the same molecule, so SAHA or Vorinostar, and also by the group of Ole Sogard with the panobinostat, which is another molecule that can be used to shock the viral reservoir. So trying to find a good molecule to shock the viral reservoir is quite complicated because latency is driven by many different mechanisms. And even if you remove one of the brakes that contribute to HIV latency, that might not be enough. So I stole this picture from a review from Doug Richmond, and it basically, I just added the names of the molecules you can use to do that. It summarizes all the molecular mechanisms that contribute to HIV persistence. So first, you have an issue with the structure of the chromatin. So basically, the chromatin is pretty packed around the LTR, so the HIV promoter, which prevent the transcription of the viral genes. In addition to that, you don't have high levels of PTFB, which is a master regulator of elongation for HIV in those resting CD4 T cells. Also, you might miss some essential transcriptional factors for HIV, such as NF-kappa B, I don't know where it is here, but it's somewhere on the slide, or NFAT-C. And the cool thing is that you can use different molecules to try to trigger those pathways and force viral gene expression. So for the chromatin, you can use the HDAC inhibitors that I just mentioned in the 3 in vivo studies. You can use PKC agonists to force NF-kappa B expression or transfer into the nucleus. Also, those bromodomain inhibitors have the ability to activate PTEF-B, and those cytokines might also induce viral gene expression by phosphorylating STAT, which might in turn reactivate the latent reservoir. So at this stage, I'd like to come back to my previous slide and this idea that all the memory cells are not the same. And why is it important? I think it's important in those track and kill strategies for the following reason. So we did a very simple experiment in which Rémi Fromentin in the lab sorted central memory, transitional memory, and effector memory CD4 T cells and exposed those sorted subsets to those candidates molecules that could reactivate the latent virus. And we'd use RNA sequencing to identify the genes that are modulated in those different subsets. And what you see on this picture, and I will skip the CD3, CD28, which obviously will never be used in patients, but the prostratin and the vorinostat, which are two molecules that we would like to use in shock approaches, as you can see, they induce genes in some subset and not in others. So if you look at the most dramatic example is probably the vorinostat or SAHA, only 3 genes are commonly modulated between central memory, transitional memory, and effector memory. So that means that when you put this molecule in a patient, it will act in a completely different ways in the 3 major reservoirs for HIV, suggesting that these molecules might act on some of the reservoir, but not on others. And we are actually doing studies right now to demonstrate whether this is true or not. I want to finish with some words about immunological strategies to eradicate the virus. Sharon is going to talk later on about the chemokine approaches, and I will talk about the negative regulators. So I guess most of you know PD-1, which negatively regulates T-cell responses. PD-1 has two known ligands, PD-L1 and PD-L2, which are mostly expressed by myeloid cells. And if you block the interaction between PD-1 and its ligand, you can restore T-cell proliferation and secretion of cytokines. So we were interested in PD-1 for a reason that is too long to mention, but just by chance when we were working with Lydie Trotman, who published this study back in 2006, she was able to show that putting the blocking anti-PD-1 antibody in a culture from a viremic donor, you increase T-cell proliferation and cytokine production. And I kept the supernatant and did a P24 ELISA to measure the virus release. And the hypothesis was, well, if you block PD-1, you restore T-cell function in this culture, you will kill more infected cells and get less P24 at the end of the day. And we ran the ELISA and saw exactly the opposite. So basically, when we added the anti-PD-1 antibody to the culture, we increased the level of viral production in those cultures, which was pretty hard to understand to us. So we then asked the question whether or not PD-1 positive cells were enriched for HIV, and these data were generated in untreated HIV-infected individuals, and we sorted central memory, transitional memory, and effector memory cells based on their PD-1 expression, measured integrated DNA, and we were able to show that yes, PD-1 positive cells are enriched for the virus. And more importantly, if you look at the capacity of these cells to produce virus, again, these are 4 different donors, we found that the PD-1 positive fraction produced much more viral particles than the PD-1 negative fraction. So that was pretty cool to identify a marker for HIV, for infection in viremic patients, but the next question was, is there any role or functional role for PD-1 at the surface of these cells that are preferentially infected? So what we did, and Sandrina da Fonseca performed this experiment, we got CD4 T cells again from untreated HIV-infected donors, and when you put them in culture, they produce some virus, and this is by P24 as a percentage of the maximum. And if you stimulate them with CD3, CD28, obviously they will produce much more virus. And interestingly enough, If you engage PD-1 with its ligand PD-L1, you can abrogate this enhancement of HIV replication. And Sandrina was able to show that this is happening at the level, at the transcriptional level. She transfected CD4 cells with an LTR luciferase reporter, and she was able to show again that when you engage PD-L1, or PD-1 with its ligand PD-L1, you can abrogate or reduce the transcriptional activity at the LTR. So what's happening during antiretroviral therapy? The first thing we observed is that PD-1 is not fully normalized, even though it's pretty close to the values you will see in seronegative subjects. This difference is still significant. And of course, antiretroviral therapy also reduces activation, so reduces the expression of PD-1. More importantly, there was this nice correlation, positive correlation between the size of the reservoir, measured by integrated DNA, and the level of PD-1 expression. So Remy and others and me and many people in the lab sorted cells from a large number of virally suppressed individuals to determine whether or not those PD-1 positive cells are also enriched in fully suppressed individual. And yes, it's true in central memory cells, it's true in transitional memory cells, it's not true in every single patient, as you can see, it's a kind of a mix, and it's definitely not true in effector memory cells. But if you sort those cells and then you look at the functional reservoir, so the reservoir that can be induced, and we use TILDA, the assay I described earlier, then we can see that in every single patient we've looked at, yes, there are more cells with inducible HIV in PD-1 positive cells as compared to PD-1 negative cells. What I think was really impressive, or I would say important to us, is the fact that if you reactivate the latent reservoir in those virally suppressed individuals with CD3, CD28, as soon as you engage PD-1 with its ligand, there is no reactivation anymore, again suggesting that PD-1 can be a brake for viral activation and can indeed contribute to the persistence of these latently infected cells during antiretroviral therapy. Because I don't want to be over time, I will skip this slide just to tell you that this is an horrible experiment that Remy designed, horrible in the sense that it takes weeks and thousands of dollars, but we wanted to be able to demonstrate whether or not blocking PD-1 could have an effect on the latent reservoir. The first thing we measured is the cytokine production, and as expected, and even in those virally suppressed subjects, the anti-PD-1 antibody was able to increase a little bit the secretion of TNF-alpha and IL-2. This is done in a system that is not optimized at all, I mean, not ideal, I would say, because we're using SE and SCB, and obviously we want to use HIV peptides to show that, like Dan Kaufman did previously. And finally, yes, it seems that the anti-PD-1 antibody can lead to some reactivation of the viral reservoir, or I should say enhancement in the viral production from the viral reservoir. This is the mock control, this is the blocking anti-PD-1 antibody, and this is the isotype control. So in these 7 donors, we saw a significant effect of the blocking anti-PD-1 antibody, suggesting that it could be used to shock the viral reservoir. PD-1 is not the only one. There are many other negative regulators that have been described. And again, in collaboration with Remy, but also with Sharon, with Steve Dix at UCSF and all the people there, we measured 8 negative regulators and correlate them with the size of the reservoir. We identified 2 new negative regulators, LAG3 and TIGIT. And when Remy did the sorts on these different molecules, he was able to show that the more a CD4 T cell will express these negative regulators, the more it's going to be enriched for the viral reservoir. So again, that suggests that those negative regulators play an important role in HIV persistence during antiretroviral therapy. So very briefly, some conclusions. So those 3 subsets, central memory, transitional memory, and effector memory, are the major reservoir for HIV during ART. By treating very early on at the FEV1 stage, you can prevent the establishment, or at least reduce the establishment and maintenance of this long-term viral reservoir. I hope I convinced you that PD-1 is a marker of infected cells, both off and on ART. By blocking PD-1, you can increase cytokine production, but also viral release, even though the system we've used so far is quite artificial, and that other negative regulators, such as LAG3 and TIGIT, may have similar effects. So finally, I'd like to thank all the people who have contributed to this work. More importantly, Rémi, who has done probably 90% of the work I showed today. Other people, Marion has joined the lab. She's a PhD student, and she's working on the molecular mechanisms of HIV latency. And Amélie just also joined the lab and will run mini Tilda for the team within the next few months. Our colleagues at VGTI Florida, of course, Andrina da Fonseca and Francisco has developed Tilda. Rafik has been involved in many of these studies. Our colleagues from MHRP for the Thai study, from Merck, McGill, Jean-Pierre, from UCSF, of course, Steve Dix has been involved in this. Sharon has been a great collaborator for the past 5 years in this study funded by the NIH. And finally, I'd like to thank the study participants. Thank you very much.